Hello and welcome to Pale Blue Thoughts once again. We have been continuing with the series explaining how modern medicine works and also explaining the working principles of woo-woo medicines or the so-called complementary and alternate medicines. Now we have been taking various medicines in our lives. Let me ask you a few questions. How many of you know how these are developed? How do they act within the body? What happens within the body when you take a medicine? Why do we have side effects? And how does the tablet or pill know that I have a pain in my leg or my head? Suppose you have a disease in your kidney and you are eating a pill which reaches your stomach. How does it then work its way to the kidney and cure the disease? What is the mechanism that enables a pill to work on specific areas of the body? If you have not been able to answer the above questions, stay on, as Pale Blue Thoughts is going to explain the actions of modern medicines within the body. You all know that the human body is made up of cells. We have billions of them. Each have their own function and they work individually or collectively. Together they form tissues that themselves form organs and eventually the entire body. For example, a normal liver has around 240 billion cells in it. Each of these cells are made up of nucleic acids, proteins, carbohydrates and lipids. Of these, let us concentrate on proteins first. Proteins are large complex molecules that play many critical roles in the body. They do most of the work in cells and are required for the structure, function and regulation of the body's tissues and organs. Some of the most common proteins and their functions are antibody. We all know what it is by now thanks to COVID. Antibody cell proteins bind to specific foreign particles such as viruses and bacteria to help protect the body. Then there are proteins called as receptor proteins. Again, taking the example of COVID, you may have heard that the coronavirus uses its spike proteins to attach itself to the receptor proteins within our cells. These are often found on the surface of the cells and is often used for binding to different molecules. Then we have enzymes. Enzymes carry out almost all of the thousands of chemical reactions that take place in cells. They also assist with the formulation of new molecules. The chemical reactions that keep us alive, our metabolism, rely on the work that enzymes carry out. Messenger proteins. Messenger proteins, such as some type of hormones, transmit signals to coordinate biological processes between different cells, tissues and organs. Our growth hormone, the chemical that causes us to grow, is a perfect example. Then there are structural component proteins. These proteins provide structure and support for cells. Example are our muscles. On a larger scale, they also allow the body to move. Transport or storage proteins. These proteins bind and carry atoms and small molecules within cells and throughout the body. The substances transported by these proteins can include ions such as sodium and potassium, sugars such as glucose and many more. Proteins are made up of hundreds or thousands of smaller units called amino acids, which are attached to one another in long chains. There are 20 different types of amino acids that can be combined to make a protein. The sequence of amino acids determines each protein's unique three-dimensional structure and its specific function. Since this sequence determines the function of the proteins and structure, any change causes a change in the proteins or amino acids causes a change in its structure and function. So I hope you have a fair understanding of proteins by now. Now let us see what is a disease. A disease is a particular abnormal condition that negatively affects the structure or function of all or part of an organism, and that is not due to any immediate external injury. Diseases are often known to be medical conditions that are associated with specific signs and symptoms. there are two main reasons or channels for getting a disease. One is caused by an external source like the entry of a virus, bacteria, fungus, and so on. Taking a recent example, the SARS-CoV-2 spike binds to its receptor human ACE2 through its now famous spike protein. Then it enters the cell and starts to replicate and causes the cell to change its normal function. Then we say we are infected. The next cause is internal. As I mentioned, every protein has a definite structure. Sometimes there might be a change in the structure due to various reasons. One of the major reasons is mutations. Many cancers are a result of such mutations. Sometimes there is a change in the function of those proteins. These changes to the function may decrease or it may increase. Whatever the situation, any change in the structure or function of these proteins are harmful for the body and we get diseased. Now let us see what medicines need to do to the external sources. Whenever there is an entry of a foreign body, the body tries to remove it using its immune system using proteins called antibodies. This process is what manifests as diseases. It is the body's reaction against the external source such as a virus. That is the reason why it is not a good idea to try and boost your immunity. That concept is not right and should be discarded by all of you. If your immunity increases, the antibodies would start to attack our own bodies, which results in autoimmune diseases. So what should the medicine do in such an external attack? Simply put, the medicine should try and kill the external source or it should disable it, be it a virus or bacteria or fungus or whatever. Just like our bodies are made of proteins, the body of the germ also is made of proteins which do specific functions which supports its life. So if we are able to inhibit the function of this protein using some chemical, we would be able to obstruct their action. For example, penicillin or cephalosporins are very well known antibiotics which act on bacteria. So what these do is it inhibits or destroys the cell membranes of these bacteria, much like what soap does to the coronavirus. When the cell walls are destroyed, the bacteria gets destroyed. Another way is to stop it from multiplying. This is usually done by inhibiting an enzyme which is required for the organism to multiply. So what we are going to aim at is called the target of the medicine. A biological target is anything within a living organism to which the chemical in the drug binds or is directed, resulting in a change in its behavior or function. The medicine has to literally bind itself with the target protein among the billions of proteins that are there in the body. So, the first step in medicine development is to find the exact chemical which would bind with the target protein in order to destroy it. That is called as target identification in medicine manufacturing. For this, we extract the specific protein from the virus or bacteria in the case of external sources, or the mutated protein within our body in the case of internal source of disease. There are many methods that are used to do this, but generally it is the identification of the right chemical that we require to hit the target. But we have millions of chemicals to play with. Identification of one among the millions is the toughest task. The chemicals may be plant-based as was done in the early years of chemistry, but these days we have a structure in place where hundreds of thousands of chemicals and their effects have been identified and recorded and stored in chemical libraries. Even today, there are some plant-based chemical extractions done to obtain certain chemicals, but they do not follow the crude method of taking and using an entire part of the plant as done in Ayurveda and other herbal medicines. Only the required chemical is extracted and purified to remove any contaminants or toxic materials like heavy metals which may be present in plants. Today, since chemistry has developed so much, we can create hundreds of thousands of chemicals with literal ease. Now the chemicals are screened against the target protein to see which one binds to it and can produce a neutralizing or destroying effect. With today's technology, we don't even need to screen each chemical or compound against the target. We can use computers to do this with ease. This is called computer-aided drug discovery. The computer can discover, analyze, and develop the exact chemical that can make the hit faster than we can. Sometimes we might get hundreds of compounds or chemicals that bind with this particular protein. We will continue screening until we find the best match. We call it as a hit. So once we make the hit, that is, identified the exact protein which can bind and produce the required effect that we need, we will introduce the proteins in an in vitro condition, which is basically in a test tube or a petri dish, and watch the action under microscopes or other observatory techniques. We most of the times may have to tinker with the chemicals to enhance its potency, and scientists can do that by making the necessary changes to the proteins using the sophisticated techniques available in modern science. Sometimes we might have to tinker with the proteins as they might cause cross-reactivity. The hit chemical might react with other proteins and cause undesirable effects to them. This is called off-target binding. So some amount of tinkering might be required to the amino acids so that we can enhance the potency and also reduce the off-target binding properties. So far, we have now isolated and identified the right chemical or protein that can hit our target. Next, we need to identify the route of administration, whether the drug has to be given through the mouth or intravenously through injections or intermuscular or through a smear. There are some chemicals that cannot be taken like food, as it would reach the stomach and the digestive enzymes would turn it into some other substance and the drug won't reach the target. In such cases, we might have to resort to injections or administer the drug intravenously. Once that has been identified, then we move on to preclinical trials. For this, we usually administer the drug on lab animals such as rat, mice, donkeys, rabbits, etc. The reason why we do this is to ensure that there are no harsh adverse effects or safety issues before it gets trialed in humans. For those with ethical concerns about animal cruelty, please note that even before applying it on animals, the mechanism of action of these chemicals are identified, and only after approvals are they administered on animals. This literally means that we are not simply injecting poison into the animals in a preclinical trial. The dose is also determined before we move on to clinical trials. Clinical trials usually come in minimum of 3 stages, termed as phase 1, phase 2 and phase 3. In phase 1, the drug is administered in healthy individuals and is mainly done to identify any toxic effects on people. Phase 1 trials are usually done on less than 100 individuals. This phase aims to figure out the highest dose humans can take without serious side effects. Investigators monitor participants very closely to see how their bodies react to the medication during this phase. While preclinical research usually provides some general information about dosing, the effects of a medication on the human body can be unpredictable. Around 70% of all drugs pass this phase. Phase 2 of a clinical trial involves several hundred participants who are living with the condition that the new medication is meant to treat. They are usually given the same dose that was found to be safe in the previous phase. Investigators monitor participants for several months or years to see how effective the medication is and to gather more information about any side effects that it might cause. While phase 2 involves more participants than the earlier phase, it is still not large enough to demonstrate the overall safety of a medication. However, the data collected during this phase helps investigators come up with methods for conducting phase 3. On an average, only 33% of medications pass phase 2 and move on to the next phase. Phase 3 of a clinical trial usually involves up to several thousands of participants who have the condition that the new medication is meant to treat. Trials in this phase can last for several years. The purpose of phase 3 is to evaluate how the new medication works in comparison to existing medications for the same condition. To move forward with the trial, investigators need to prove that the medication is at least as safe and effective as existing treatment options. Phase 3 trials are usually double-blind, which means that neither the participant nor the doctor knows which medication the participant is taking. This helps to eliminate bias when interpreting results. Also, placebos might be introduced and the group is randomized to eliminate further biases. This method, called the randomized placebo-controlled double-blinded studies, have been discussed before on this channel. Now all the test reports and results right from protein identification right up to the results of phase 3 trials are sent to a drug approving authority like the US FDA. The drug control authorities scrutinize each of these reports and results meticulously before deciding to approve or reject the medicine. Once they are satisfied that all the above said methods and phases have been completed satisfactorily, the FDA approves the medicine. And then the drug comes into the market. The whole process may take 10 to 12 years on an average. Some drugs even go through a phase 4 after the approval has been done, around 30% of the cases. This can last several years as investigators use this phase to get more information about the medication's long-term safety. You may have heard of drugs being revoked or removed from market after many years. That is because of phase 4 results or because we found a better and more efficient drug for the same disease. The procedures and steps involved are much more well-defined and scientifically structured than just shaking and diluting or mixing a lot of plants and herbs into one pot and making a decoction. None of the woo-woo systems employ any preclinical trials, nor do they study the mechanism of action like it is done by modern science. For them, it is all about what is written in the ancient books. Whether it is right or wrong doesn't matter much. Today, they claim that they are also doing research. But what is the major flaw in their approach? They first release the medicines and then do a research, while in modern medicine, the research is done first and then the medicine is released in the market. All mechanism of action and toxicology and pharmacology studies are completed, approved and documented in public view before the drug reaches the market. Woo-woo medicines, on the other hand, reaches the general public without many studies, and no toxicology or long-term side effect study is done on them. They just claim to have no side effects because no one has looked for it. So which method do you think is better and ethical? You decide. And the next time you feel like going for a woo-woo system of medicine, remember what you learned through Pale Blue Thoughts. In the next part, we will see how the medicine reacts within the body and how the body reacts to a medicine. If you like this video, please hit the like button and share this for the benefit of others. Until next time, it's bye-bye from Pale Blue Thoughts.